Hello dear students, welcome to all of you. So today we're going to start with a new topic that is microbial analysis of milk. So in this particular topic we're going to see different tests that are routinely used for analysis of raw as well as pasteurized milk and milk products. Okay. So in that we're going to study methylene blue reduction test. So this test is very popular and it is known as MBRT test. So methylene blue dye reduction test for accessing raw milk quality. So in this particular test we use dye that is known as methylene blue dye. It is blue color dye, and after adding this dye to milk, so milk get change its color, and this dye get reduced. Okay, so this dye get reduced. So particularly this test is based on redox potential. Okay, so methylene blue dye reduction test, commonly known as MBRT test, is used as a quick method to access microbiological quality of raw as well as pasteurized milk. So this is quick method of analysis of microbial analysis of raw as well as pasteurized milk. So this test is based on the fact that blue color of dye solution added to milk get decolorized when the oxygen present in milk get exhausted due to microbial activity. Okay, so it is obvious that this blue color dye, when you add to milk, so this dye imparts its color to milk, and milk get blue color after addition of this dye, and due to microbial activity the oxygen in the milk get consumed by microbes and this dye get decolorized. So this is the basic principle behind this test. This test is basically depends upon redox potential, and this redox potential is depends upon microbial activity. So as soon as the decolorization happens, more inferior is the bacteriological quality of milk assumed to be. So when the milk get decolorized in shorter time, so the quality of milk is considered to be means it is inferior. Okay, so this test is widely used as a dairy at the dairy reception dock, processing unit and milk chilling centers, where it is followed as acceptance rejection criteria for raw and processed milk. So this test is very basic test used to check quality of raw as well as pasteurized milk. So grading of raw milk based on MBRT. So MBRT test may be utilized for grading of milk, which may be useful for milk processor to take a decision on further processing of milk. Okay, so as per BIS 1479 part 3 at 1977 criterion for grading of raw milk based on MBRT, so grading depends on the following chart. Okay, so procedure. So this test has to be done under sterile conditions. So what you have to do, all the operations regarding this test you have to perform in sterile conditions. So in that, first you have to take 10 ml of milk sample in sterile MBRT test tube. Then you have to add 1 ml of MBRT dye solution, which is as concentration 0.005%. Then you have to stop the tubes with sterilized rubber stoppers and carefully place them in a test tube stand dipped in surgical water bath maintained as 37 degrees Celsius. So record this time as a beginning of incubation period, and declaration is considered complete when only a faint blue ring about 5 mm persists at the top. So basically you have to check decoloration of milk in this particular test, and you have to check decoloration as the time goes. So recording of results. So how to record results in this particular test? So during incubation you have to observe color changes. So you have to just observe colors change of milk. So if any sample is decolorized on incubation for 30 minutes, record the reduction time as MBRT 30 minutes. So if your sample get decolorized within 30 minutes, so this your MBRT time is 30 minutes. So you have to record reading in such a way, then reduction time in whole hours. For example, if the color disappears between zero point 0.5 to 1.5 hours readings, then record the result as MBRT 1 hour. Similarly, if it get decolorized between 1.5 to 2.5 hours, then consider MBRT as a 2 hours. Immediately after each reading, remove and record all decolorized samples and then gently invert remaining tubes if decolorization has not yet begun. So this is a chart that is showing reading of raw milk based on MBRT. So after addition of dye, if you kept the milk for 5 hours and above and still it get not decolorized, or it gets started decolorizing after 5 hours and above, then this quality of milk is very good. Okay, so this quality of milk is very good. Then second criteria, if that dye sample get decolorized after 3 to 4 hours, then its milk quality is considered as good. Then if some of the samples they get decolorized after 1 to 2 hours, then that milk quality is considered as fair. And if the milk gets decolorized sooner, within half an hour, then it is considered as a poor quality of milk. So this chart shows basically the reading of raw milk based on MBRT. So good quality, very good quality of milk considered after decolorization after 5 hours and it's above. Okay. So grading of milk in MBRT test. So if the good quality of milk, when it gets reduction time is almost 5.5 an hour, so you can keep this milk up to 40 hours. Okay, you can keep this milk up to 40 hours, and the bacterial count at this good quality milk it considered below 5 lakh. Okay, below 5 lakh. Then if you consider fair quality of milk, then its reduction time is up to 2 to 5.5 hours, and you can keep this milk for 30 hours, and its microbial load is considered 5 to 40 lakhs. Then if you see the bad quality of milk, so its reduction time is 20 minutes to 2 hours. So this bad quality milk get decolorized very sooner, and you can no longer keep this milk more than 10 hours, and its microbial load is considered as an 40 lakhs to 2 crores. And if you see very bad quality of milk, so it is again decolorized very sooner, less than 20 minutes, and you cannot keep this milk up to less than 10 hours, and its microbial count is considered as an 2 crores and above. So there are some factors that affect MBRT test. So first, since the oxygen content must be used up before color disappears, any manipulation test that increases the oxygen content affect the test. Okay, so there should be no manipulation in such a way that oxygen of the milk get increased. Okay, cold milk holds more oxygen than warm milk. Okay, so pouring milk back and forth from one container to another increases the oxygen. Okay, and next, during the milking time, such as oxygen may be absorbed. So basically this test is depend upon concentration of oxygen. So concentration of oxygen after adding dye, it should not be vary, okay, by this some external factors. Okay, so the rate of reduction of dye depends upon the types of microorganisms. Obviously the rate of reduction of dye is depend upon different types of microorganisms, as the oxygen utilizing capacity of different microbes is different. So this rate of reduction of dye depends upon that which type of microorganisms they are present in milk. Okay, so coliforms appear to be the most rapidly reducing microorganisms. So in this particular group of microorganisms, coliforms they appear to be the most rapidly reducing microorganism. Then closely followed by Lactobacillus lactis species. Lact is some of the fecal streptococci and certain micrococci. Okay, so this pattern is also followed by some lactic acid bacteria like lactose, lactis species, then some micrococci and some streptococci. Cyclotrophs reduce methylene blue very slowly. So these are the type of microorganisms. They reduce this dye, particular blue dye, very slowly, and will get longer reduction time. So presence of large number of leukocytes as in mastic milk will affect the reduction. Okay, so third, the presence of large number of leukocytes as in mastitic milk will affect the reduction time materially. Okay, so if there are a large number of leukocytes due to disease mastitis, so they will again reduce the time of reduction. Okay, then light hastens reduces reduction process, and therefore the test should be carried out in relatively low light. Okay, so light basically affect this reduction of dye. So you have to carry all these procedures in relatively low light concentration or light intensity. You should perform this activity in low light intensity. So concentration of dye should be uniform as increased concentration lengthens the time of reduction. So in this particular test, you have to keep all concentration of dye in uniform way. Increasing the incubation temperature augments the activity of bacteria and therefore shortens the reduction time. So incubation time again is an important factor for reduction of this MBRT test or dye, methylene blue dye. So creaming of milk causes number of microorganisms to be removed from milk and brought to surface with the rising fat. Okay, so this factor causes variation in reduction time since the bacteria are not evenly distributed. So basically the accuracy of test is increased as reduction time shortened and decoloration more uniform if the samples are periodically inverted during incubation. So this is the basic information regarding MBRT test. So in next lecture we'll we're going to study another test, that is research urine reduction test. Thank you.